I'd like to acknowledge my co-authors on this talk. Mark Rowe and Peter Alsop are the hydrodynamics modelers that are helping us find where the larvae go. Several people from our lab are technical support in the field, Eliza Lugton, Dora Mason, Henry Vanderplug, also from our cooperative institute, Maddie Tomczak. We have geneticists working on this, Rao Shigante, Tate Algier, and Lucas Vanderbilt. I forgot to mention him. And then several summer fellows. These are students who come in and work with us for the summer to get good experience. They're smart as paint and very enthusiastic. So this is I'm going to retire in September. You can hold your condolences till afterward, but this was one of the most fun field efforts I've ever been involved with. And not so much of what we learned, but just being out in the field and trying new things, because I've been involved in a long-term study of the spatial structure of the food web, and this was different. So hopefully I'll give you a little bit of a flavor for that. This is a slide or a time series that probably many of you seen over time. It's from the USGS Great Lakes Science Center. It's the results of their trawl survey for alewife populations in Lake Michigan. And there are a couple of things to point out. One is the long-term decline in the population as measured by the trawl. And then two is these peaks in abundance that just happen every so often. This one is after the 1998 year class, and this one is in 2013, following the 2010 year class. Very high survival of events in the first year of life. And the question is, what is causing this? This is a time series normalized to standardized to 0, so 0 means an average. I didn't put the 0 on there, but it's indicated by the dotted line. And then the relative to low or high, above the line is higher recruitment, below the line is lower recruitment. This was done by Les Warren, who's a student looking at early life survival of alewife in Lake Michigan, and trying to tease out what are the factors that are driving this. And so I stole this slide for him. I just wanted to point this out because it indicates the variance in survival in the first year as indexed by the abundance of alewife and trawls, or measured by acoustics at the end of the year. And then also indicate that big year class in 1998 and another one in 2010 can drive the population forward over time. The other thing to note is this variance seems to have increased in more recent times since coincident with the great explosion of quagga mussels, which we know has changed the light environment, the prey environment for young fish in terms of the density of zooplankton going down and the quality of zooplankton changing. So we've been looking at larval fish on a pretty regular basis as part of our studies of the lower food web since 2010. And so we have a measure of a larval density off of Muskegon, southeast Lake Michigan, in a really good survival year as indexed in the fall. One that's in sort of lower than normal year class in 2015, and here's 2021, which should show up this year in the trawl survey. And you can see that there's not a match with the survival to the fall between what we're measuring off of Muskegon for the really good year class and then one for the not so good year class. And that could be for a couple of reasons. It could be that the larval densities don't index year class strength. Could be where we are in the lake doesn't help in index year class strength. Or it could be things that are occurring to larvae during or after we sample them. So where you are in the lake really makes a difference, I think. Here we are on the eastern side of the lake, and this shows an upwelling event. This is output from a hydrodynamics model, but you can see the direction of flow because from winds from the north blowing the surface water south and then off from the shoreline and allowing the cold water underneath to come up. And if that cold water gets to about 15 degrees Fahrenheit or 10 degrees C, it'll kill alewife larvae. That's been documented in the past, in the lab actually. And on the west side of the lake, life is beautiful when these events come through. And on the western side of Lake Michigan, it's subject to sort of persistent, more stable upwelling and thought to push larvae offshore along with their prey environment. On the left panel, I'm just showing the results of drifter tracks that were released in 2015, showing that in the subsurface drifter, it will actually go offshore with the water. So we tried out a few new things in 2015, or it's 2023, just last summer. We asked the current modelers to tell us where water is going and where larvae would be going, if larvae are passive particles, which we think they are because they can't swim against the current. We have this geneticist in our lab now who can use environmental DNA and RNA to detect the presence and absence of larvae and maybe their relative abundance. And then Steve Ruberg gave a really good talk yesterday talking about new uncrewed vessels that have sensors on them and also fisheries acoustics and also high frequency acoustics that can measure zooplankton biomass. And we think this eventually will help us understand what the predator and prey environment is for young fish out in Lake Michigan. So what we did was we went out and sampled using our traditional nets. So I'm going to hang up my net this fall and transfer it to Steve Podhoven, where he's going to continue to monitor larval fish density off of Muskegon. And then this is an image of hydrodynamics model output. I'm going to get into details of that, but just the particles represent potential locations of fish larvae after they're released. So first, to talk a little bit about eDNA. The advantages of using eDNA are that it's well established. It can detect presence or absence of species. It can detect the differences between congeneric species like whitefish and cisco very quickly and accurately. It's a question still whether it can detect the relative abundance of species. And there are some limitations. We don't know if we know there are alewife in the nearshore zone, and we know that adults and larvae, and we don't know if eDNA can detect the difference between larvae and adults of the same species. eDNA can stay in the lake environment for weeks after the organism is not present, or if the organism is dead. So that's sort of a wrinkle in this technique. So in the lab, Rao and his colleagues did some experiments to look at how long eDNA and eRNA would last. They set up replicate tanks, 5-gallon tanks with 0 larvae, with 3 larvae, and 100 larvae of yellow perch, which we got from hatcheries. And then let the larvae sit there over time, actually for, I think, 24 hours, shedding eDNA and RNA into the water. Then they took the fish out, and then they sampled the water contents for the next 17 days to look at the degradation rate of these fish, of the material from the fish. And use that to help calibrate what we're getting in the field. Okay, so this is a a little bit complicated, so bear with me. What I'm showing here is a model prediction of where the larvae would be from output from the hydrodynamic model. So here we are at Muskegon, where our field station is. And what we first did was go offshore the first day of our cruises, late June and late July. We did 2 week-long cruises. And we sampled from near shore to offshore until we hit a patch of larvae. That was the first day of the cruise. Then when we found larvae, we did a lot more sampling. We sampled the zooplankton, we sampled the water quality, chlorophyll temperature. We took water for eDNA and eRNA measurements, and then we radioed position back to, or phoned, cell phone back to Mark Rowe back at our lab, who was ready to then run his hydrodynamics model forward in time. Because you get a wind forecast from the Weather Bureau, and the current movements are all wind generated in Lake Michigan. So he can tell us where that blob, as we called it, is going to be the next day at different times of day. And so it allowed us to repetitively sample that blob. And this blob, if we look at it, there's an area of that we call the centroid, where that's the area of higher probability, if you're a passive particle like a larvae, that you're going to be at a certain point in time. And then there's a couple of concentric areas around there. There's the 1% area, which is encircling right here in this image. So that's the 1% probability that a larvae or a passive particle is going to be within there. And then a 0.01% probability that if you're a larva, that you're going to be, if you're sampled within that area, you're going to be found there. And then outside the blob is we were just sampling to make sure that the larvae are not there as predicted by the model. So when we did a survey, we took the center point of this high probability area. We always sampled that. We always sampled within this 0.1% probability, and then we sampled outside on subsequent days and times. All right, and we looked for alewife and yellow perch larvae. It's not going. Okay. So the first day we went on the first cruise, this is late June is when alewife normally spawn, at least in our area of the lake, when the temperature gets above 15 degrees C, which, I don't know, is that 55 degrees Fahrenheit, something like that. So anyway, it was getting up there, and we found larvae, actually, that were there. And then the wind blew up about a few hours into our cruise, and the temperature dropped, and it chased us off the lake, but not before we caught some fish larvae, some alewife larvae. And then we didn't get back out until 2 days later, the 28th of June. And we were able to sample on the 28th, the 29th, and the 30th. Just anecdotally, I won't talk about this, but the stable temperatures later in the month, we found, sampling at the end of July, we found quite a few alewife larvae and tried to chase them around. Okay, here's the results of our first survey. Here's the density on that first few hours that we caught, 10.5 larvae per 1,000 cubic meters of water filtered. Their mean length was 8.9 millimeters. And sort of the average of the 2 days we were able to get out was, it dropped by about an order of magnitude down to almost 0.1 per 1,000 meter cube. And they had grown a little bit, about 0.5 millimeter, which is kind of around the growth of larvae that we're seeing on a daily basis since the mussels have gotten in the lake. It's a lower growth rate. Also, I won't show you, but we didn't really think this gave us some confidence that we're following that patch of larvae. We didn't have that confidence from when we caught a lot of them later in July. So here's the blob. Here's the centroid. So the stars indicate we caught alewife at that site in nets, and the green means we also got a positive hit for their eDNA. A circle indicates we didn't catch them there, and the red indicates we also didn't find eDNA. So if you look at right in the middle of the blob, we got larvae in the nets and we got a hit for eDNA. At the other locations, we got a few more of those situations. We got one where we got no larvae and no eDNA, but the eDNA is telling us that there were larvae, or maybe adults, outside of this blob. So encouraging, not definitive. We also did this, looked for yellow perch, and yellow perch, especially that time of year, is relatively infrequent. And for most of the areas that we sampled, we caught no larvae and there was no eDNA. There was one place we caught them and there was eDNA, and another place we caught them and there was no eDNA. Yellow perch was in very low abundance, and we still think that we're not sure what's going on there. So that's the eDNA story. We think it's promising, and we're going to repeat it this summer. So now the technology, and Steve showed this yesterday, so I'm just going to go through it pretty quickly, but there's all sorts of gizmos that he's throwing out there, and they complement each other in terms of their length of time they can be out in the environment and what they're actually measuring. But the data I'm going to show you next is from, and he showed it yesterday, is this Triton AUV. This is their longer range AUV that actually was out along the transect when we were out there sampling, and it recorded data on fish biomass, which is pretty cool. So again, for those of you who weren't here yesterday, this AUV is flying along, and it has an upward-looking transducer to measure fish biomass and a downward-looking transducer. And this is during the nighttime. And for those of you who do this acoustic sampling routinely, the fish are moving around from night to day or day to night. They usually come up a little bit in the water column, and that's what we're seeing right here. These are, Doran's telling me these are fish targets. This red stuff up here are bubbles because it's so wavy and a lot of turbulence in the water column, and relatively low densities near the bottom. And this is in about 15 to 20 meters of water, so near where we were. And this is the next day. And again, the fish looking downward, they're lower in the water column. And this is true of alewife larvae too. We know that they move up and down in the water column depending on the time of day, down during day and up during night. And we think this may be a source of error for our current models, because the hydrodynamics, the current models just assume that you're passive, you're up in the water column, and you don't move around. But fish larvae, even these tiny alewife, they can do it. They can, at least vertically, they can change their position in the water column. And then this is again at night, the night of the 27th. We're still not out on the lake sampling, but the fish are back up again. So, and I'm way out of time, I guess, right? So sorry. Yada, yada, yada. So anyway, we're going to repeat this summer. We think that the use of this additional technology, we also had satellite imagery to tell us about the water clarity and water quality. There was a guy flying an airplane looking at the the transmittance ability of the water column where we were. But we're very excited to try out these new technologies and see what they can tell us about the fate of and the survival of larvae over greater temporal spatial scales that we can actually get to during a normal survey. To sum up, we looked at the advection and the fate of larval fish. We think that can tell us something about recruitment. eDNA looks very promising to look at the presence and absence and perhaps the relative abundance. And then with the gliders, the AUVs, and the current models can all assist us in sampling and mapping what helps support fish growth or survival in these Great Lakes. That's all I've got, and thank you for your patience.